Hey everyone, welcome back. Well, welcome, I guess, for the first time to developmental biology. If you've taken my bioinformatics classes, you can see the format's a little similar, but I'm reworking this class. I may have told you in person since this is an in-person class, but I'm trying some different stuff here. So I'm reorganized the way we go through the content, tried to trim some stuff, make it a little more interactive, etc. So we're going to start today with a quick reminder of meiosis. And so this image is not a scientific image at all. Well, I guess in a sense it is. So this is what, if you ask ChatGPT to give you an image of meiosis, and so you can see it's just kind of, it's cool. It's fun to look at. It looks like it's made out of yarn or something. It's kind of weird, but I like to kind of mess around with the AI stuff. So so my title slides are kind of going to be generally what ChatGPT thinks or what AI thinks some of the science stuff is. Okay, so we want to just kind of have a brief reintroduction, I guess, or primer. Maybe have been a little while since you took molecular or genetics or intro bio or any of those classes. So let's quick go through the phases of meiosis. So remember, the difference between mitosis and meiosis is that meiosis ends up with haploid cells, or cells that have only half the genetic information. And this is on purpose, right? Because if you are going to create an offspring of some sort, you want to take half from your mom, half from your dad, or half from one parent, half from the other parent, right? And create a whole individual. If you didn't go through these phases, what happens is you end up with doubling your genome each time, right? So if you were, say, sperm and egg were created from mitosis, where you have a full 2N complement of the genome, then if they come together, there's going to be 4N. And then if those people have offspring, then it'll be 8N, and it'll just keep doubling each time, right? So we don't want that. So remember, in prophase, the chromosomes condense into the actual, shape that you see like in a karyotype, instead of being all unspooled and able to be transcribed. So they condense into these. Let's see if I can turn on my pointer here. All right, so they condense into these full chromosomes. So the pair of the homologous chromosomes line up along the equator of the cell, and they get pulled apart by these spindle fibers to each opposite pole of the cell. Then we start to get this, during telophase, we start to get this cleavage point where the cells break into two. And so then we have single pairs, right? So this would be normally where like mitosis sort of ends. And so, but now we have another forming of spindle fibers and another realignment. And instead of pulling apart the pairs that we had before, it pulls apart the two halves of each chromosome here, right? And so now we end up with half of the genetic information in each of these daughter cells at the end, okay? And so like I said, let me get my cursor. It's gonna be kind of weird working with this setup here. So as I said, this is really important for the formation of eggs and sperm, and to create these gametes, right? And so we have spermatogenesis and oogenesis, so the formation of the egg. And remember, when we'll go through the actual process of these a little more in detail, but I just wanted to kind of make a reminder, give you a reminder that these are the products of this meiotic divisions. And so in spermagenesis, remember, we start with 2N, so 2 copies of each chromosome, and we end up with 4 daughter cells that are each a single sperm, so a single 1N haploid. The egg formation is a little different because we get these polar bodies, and so we don't end up with 4 daughter cells, because remember, when you're making for sperm, there's millions and billions of sperm are made in most male individuals, at least in humans, that we kind of focus on in this class. But eggs, there's a lot less, more limited supply, and so part of that is theoretically, part of that is because of the upkeep required for eggs. So women generally have them eggs from a very, very young age and keep them in their body and release them sporadically throughout their life, and they're not creating them constantly. And they're just more to them, and there's more energy involved with upkeeping them. So theoretically, that's the explanation that I've seen for why we don't have billions of eggs. And they're bigger too. They're way bigger than sperm. Obviously, sperm fits inside or binds to the cell of an egg or cell wall of an egg. We'll get into that process too. Anyways, getting back off my tangent here. So I just want to remind you that this is the product of Meiosis. Quick primer. Reminder, there's plenty of really good YouTube videos that if you want to get into the real nitty-gritty of it, but I just wanted to refresh your memory because you should have learned this already in genetics. But that is it for this real quick primer video, and I will catch you in the next one.